Yeah, so in synthetic biology this semester, we had fun thinking about two new problems. Well, I had fun at least. Well, we'll see if everyone else had fun. But two new problems that we think we can solve with synthetic biology and using new techniques in biology and engineering new systems. So anyway, the students made great progress, and Lindsay and Charlotte are going to explain those problems and our solutions that we worked on. All right.
All right, so hi, my name is Lindsay Newkirk, and I'm presenting with Charlotte Sprayson on the spring 2022 semester of MCDB 3000, Engineering the Soybean to Produce Squalene and Bovine Milk. So my group was responsible for producing cow milk and soybeans. And so some of the reason why we may be doing this is because dairy cows are constantly mistreated and abused. They are hooked up to these milking machines, and they're unable to roam around freely, and they're constantly forced to be pregnant so that they keep on producing milk. Being able to harvest cow milk from soybeans would be a more ethical alternative to obtaining milk, because you wouldn't need to use cows at all. It could be as simple as growing, harvesting, and processing a crop. So the first thing that we needed to do was identify what component of milk that we wanted to make. So we chose casein, which is the most abundant. It comprises about 80% of the total components of milk. It forms a protein micelle with 4 different subunits, which are the alpha S1 casein, alpha S2 casein, beta casein, and then kappa casein around the outside. We also identified a kinase called FAM20C, which phosphorylates the different individual subunits in order to form a proper micelle. So for our experimental design, we first identified the coding sequences of all 5 of these genes that we wanted to insert. We took these genes, we optimized the codons for soybeans, and then added an export signal so they would be exported into the periplasm. So it's easier to extract them. After we had our completed sequences, we sent them off to a company named Twist, who synthesized them for us and sent them back. And then to assemble the genes into plasmids, we used a process called Goldengate, which I'll go into in a minute, which uses type 2 restriction enzymes and sticky ends to insert the genes into plasmids. After we got our genes into their own vectors, we added a promoter and terminator. And as of right now, this is the stage that we got to, and we are in the middle of assembling all of the promoter and gene and terminator constructs into the full overall plasmid. Our next steps would be to transfect this into an agrobacterium, which we could spray onto soybeans, and it would inject our plasmids into the soybeans, and we could grow them up. And through a few rounds of selection, we would have homozygous soybean plants that we could extract the cow milk from. And so going into how Goldengate works, we use Goldengate for all of our gene assemblies. And so Goldengate uses type 2 restriction enzymes to do so. So how you do it is you have a transgene, and you combine that with a vector, which contains ampicillin resistance and a blue-white selection gene to be cut out. The type 2 restriction enzymes are key here because they bind to one part of the DNA, but they cut a few base pairs away. So it leaves these sticky ends that you can program to insert your genes in a certain order. The restriction enzymes will cut out the blue-white selection gene, and the transgene will come into the vector, and the sticky ends will anneal, and a ligase can ligate the NIC, so you'll have a full transgene plasmid. The restriction enzyme binding sites will leave with the blue-white selection marker. So the restriction enzymes cannot cut out the transgene after it's been inserted. After you have done this process, you're left with a plasmid with your transgene and the ampicillin resistance. This will get transfected into E. coli and plated on an ampicillin plate so that only your gene constructs with the ampicillin resistance can grow. You can then isolate the white colonies from this DNA because the blue colonies show that the blue-white selection gene did not leave, meaning that that's not a good colony for us to use. So we can then isolate that DNA and purify it and then repeat the process. When you repeat the process, you'll be putting it into a different resistant vector. So you can check and make sure that as you go along, you are only using your new gene constructs that you've made. And we also switch restriction enzymes so that you cut out in different directions. And so here we have some of our results. This is our restriction enzyme digest from our one of our assemblies of our promoter gene terminator constructs. Here you can see this is the vector backbone that was cut out. It's all of them are at 1580 base pairs. And then here above and below, you can see the individual gene promoter and terminator constructs. Here we have an unsuccessful colony because you do not see our promoter and gene terminator constructs. So we know that would be a colony that we would not want to use. And now I'm going to pass it on to Charlotte to talk about squalene.
Hi, yes. So squalene is a high value chemical molecule that currently we use in a lot of skincare and vaccine products. But the current source, so the main source of squalene, is actually from the livers of sharks. So lots of sharks are fished out of our waters every year to be killed and have their livers extracted for these fatty lipids, which include squalene, for use in all these sorts of products. So squalene itself is actually expressed in most eukaryotic organisms, just not as it's not as concentrated as of that in the livers of sharks. However, we believe if we can modify the squalene pathway, we can increase squalene in any eukaryotic organism. And of course, in this case, we are using soybeans. So yeah, move on to the pathway. So yes, squalene is made in the mevalonate pathway, as you can see here. But squalene is synthesized by squalene synthase. So in order to try and increase the amount of squalene we're producing, we want to basically upregulate its synthesis by adding an extra copy number of squalene synthase. So we did a bit of research and we found that ginseng, another herbal plant, has a particularly effective squalene synthase. So we took the code for that and had that gene synthesized by twist. We also want to make sure that our squalene isn't being then produced into the downstream metabolites, the sterol, which are basically going to become its hormones. We don't want squalene being used up for that. So we want to block the next enzyme in the pathway, which is squalene epoxidase. And we want to achieve this by knocking out its mRNA so it can't be expressed into the enzyme with an RNAi. So yeah, moving on to RNAi. So of course, as I said, with the squalene synthase, it was very simple. You just got your gene synthesized. With the RNAi, it was a hell of a lot more complex. So in terms of the squalene epoxidase, there are 7 different paralogs of this enzyme or / gene, which basically means there are 7 genetically different versions. Now, RNAi is very precise. It needs to have this 20 base pairs in the fragment perfectly complementary to the mRNA it's trying to knock out. But none of these, when doing a multiple sequence alignment with these 7 paralogs, there is not a single 20 base pairs like that. So you can't just make one, sequence as your RNA. You have to take a different part of each paralog. So with this multiple sequence alignment, we took 200 base pairs from different sections of each paralog and then constructed them into this RNAi. So you have all your 7 paralogs, a bit of strand for the loop, and then all the paralogs again. And this is so when the template strand is, what's it called? Transcribed, there we go, into mRNA. This mRNA can then fold back on itself, anneal, and form that lovely little hairpin, which the dicer complex comes and chops up into those 20 base pair fragments I mentioned. And the RIS complex then goes and slices up any mRNA, which is complementary to the 20 base pairs that it's now holding on to. So with this, we hope to knock out all 7 paralogs, which means we'll have complete knockout of that gene. Onwards, shall we go? So once we've got our RNAi construct nicely built and our gene, the next step is simply, as Lindsay previously described, is to get them into plasmids with the Goldengate method, which she explained so fully, I probably don't need to explain to you again. But, the first step, we stuck our gene and our RNAi into a plasmid. We chose the white colonies, and then we went again and did it again with the promoter and terminator. The only point I'd like to add is that after we put it in the initial plasmid, we sent it off to get sequenced just to make sure that the gene and the RNAi construct we received from twist DNA were actually accurate and there wasn't any base pairs that had been lost in the synthesis. So what have we actually produced? So this is what we made. It's pretty simple looking at the moment. We've made our Panax ginseng, which is the name of the plant, and its squalene synthase gene with its promoter and terminator, and we stuck our RNAi construct in. So once we had made these, we wanted to just do a last minute check that they'd actually successfully been put in the plasmid. And to do this, we did a restriction digest. Yep. So here you can see that our ginseng squalene synthase is about 1,800 base pairs, and it's pretty hard to see compared to the vector, but there is actually two lines there. One is the gene along with the promoter and terminator, and the RNAi, which is far more obvious considering it's far bigger, is about 4,000 base pairs, which is obviously visible up there. So we now know that we've successfully made those constructs, isolated those plasmids, and now we have that DNA waiting for the next steps. So as Lindsay touched on, the next steps is to get this plasmid into soybeans. So the first thing I'm going to do is try and integrate these plasmids along with a GFP marker, so a green fluorescent protein marker, into agrobacterium. So now you have this agrobacterium. It's special, and it's a bacterium that can infect plants, and it can transfect some of its DNA into the plant of which it's infecting and make it express it. So we're hoping to hijack that mechanism and stick our agrobacterium all over our soybeans in hope that some areas will be transfected with our plasmid construct, which will be able to tell it's been successful by observing fluorescence of the GFP marker. Now, we then want to extract the contents of this area that's been transfected and see if it's actually a higher concentration of our desired products, be that the squalene from my team or the casein proteins from Lindsay's team. Now, if it's successful, it may not be. We can't guarantee, but if it should be successful, we now want to completely make this soybean into a valid model to produce these products consistently. So what we do want to do, we want to rebuild our construct with an inducible promoter so that it only causes the production of squalene or casein in the soybeans themselves, not the entire plant, not the leaves. We want this plant to grow up normal and healthy so it can produce tons of soybeans, and then we want those soybeans to be absolutely packed full of the good stuff. So once we've made this inducible construct, we then try the whole thing again with agrobacterium. And the hope is that eventually we will transfect part of the soybean that will become the next germline. So although this plant, as it grows up, will only have an area of it transfected, because it's the germline area that's been transfected, its progeny will all be fully hybrid with our desired construct with the inducible promoter and the ability to produce these abnormal products. At which point you take the soybeans, you make a big old field, and boom, you have squalene from soybeans or milk from soybeans. So I hope with this, I have demonstrated to you how revolutionary synthetic biology can be and the possibility of producing loads of different chemicals in a more ethically and environmentally friendly way. And with that, I'd like to say thank you to Dr. De Decker for running this class, for all the support, and of course to Simon Kalmus, who I'm not sure if he's our official TA, but has pretty much worked as one. And of course, thank you to all you guys for listening. I forgot to ask for questions, if there are any.
If there are any questions, yeah. I'm actually wondering, I have not a complicated question, but I'm curious about how you would respond to somebody who says there's no way I'm going to eat any GMOs. Do you have something prepared for dealing with that kind of thing from the court of public opinion?
As Dr. De Decker likes to say, one of my favorite things is if you isolated just the portion that you're putting into the plant, what would you have? You just have a pile of protein powder. There's not much harm going into any GMOs. It's just a sustainable way for us to increase our food supply and have the things that we still like while still making them in a more sustainable way.
Could not have said it better.
Yeah, that's a great answer.
You guys were awesome. This might be a little bit outside of the scope of your research. I was wondering why squalene is so dense in those parts of the shark. That might be, sorry if you can't have an answer to that.
No worries. I actually had this question earlier on my poster, of which I already went and constructed a nice little answer for. Effectively, sharks are non-bony fish. And bony fish have the ability that they have this basically bladder, which allows them to be buoyant, where non-bony fish like sharks do not. So instead, what they do is they have a very high concentration of fats and oils in their liver, which allows them to be more buoyant. I mean, most animals have fat and liver in their fats and oils in their liver because that's pretty much where it's processed as such. So sharks just do this to the extreme. They have a hell of a lot more fat and oils, and squalene, as it is a lipid of sorts, is also concentrated in there. And sharks are a lot bigger than maybe some small non-bony fish. You want to get your big fish, you take the big liver, and then you've got the highest concentrated amounts of fats and oils you can find, pretty much.
Awesome, thank you.
Was there a reason that you chose soybeans specifically?
Dr. Decker works with a lot of soybeans with his research as well, but soybeans are a really good plant. They don't take a lot of energy to cultivate, and they're really easy to cultivate.
Makes sense, thank you.
So there's a question that came up in the chat, and it was specifically about milk, but I think this could go for either one. How would extracting milk or maybe squalene from soybeans work?
So we exported them to the periplasm, which is the space in between the cell membrane and the cell wall. So ideally what we would do is we would put the beans in a vacuum, and this would cause the cell membrane to go outside of the cell wall, and all of the contents would be pushed out into the extracellular space. And from that, we could spin it down. And at least for milk, we're hoping that it would be in the soluble layer, and we could extract it that way.
Yeah, it's pretty much how you'd isolate any particular protein or DNA from a cell. You lyse that cell, release all the contents, and then you, depending on what you're trying to isolate, you do a series of tests. So, if it has a particular, if say squalene had a particular affinity for another chemical, if it was like to bind, you do an affinity column and try and get squalene to stick to the sides and everything else to flow through. It's really dependent on what type of protein you isolate, but there are so many different mechanisms for doing that.
You mentioned you used a mechanism in bacteria where they'll put some of their DNA into the plant's DNA and have that be expressed. Is there any way to increase the likelihood that the DNA and the genes that you put in will be what gets transferred?
I am not sure if we can adapt conditions. There might be certain conditions with agrobacteria favor sticking their DNA into things. How, for example, phages prefer to be lysogenic when there is not many resources around because they don't want to destroy their very few hosts. It might be something like that we can control. Otherwise, I believe the agrobacterium, we might be using a specific strain, which, how you can modify, you can get like particular strains of bacteria, which pretty much have nothing but the basic components they need to survive. And then when you stick your gene in, that's pretty much the only interesting gene in there. So there's a high likelihood that your gene is the one that gets moved. I think we'll be using something similar to that. So an agrobacterium strain with the very basics it needs to be able to transfer its DNA or infect plants. And then just our construct on top as a little extra to go in. I believe. Don't trust me on that.
Thank you.
And then just one last question from the chat, and because I think this is interesting, with the things that you chose, are you limited in scope because you're using soybeans, or do soybeans allow you to use an unlimited number of, or try to synthesize an unlimited number of things? In other words, how limiting is the choice of soybean?
I know you can look up a lot of different research articles. For our final, we had to write a paper on a different system for or another synthetic biology system, but there's a bunch of different compounds that you can go and produce in soybeans, yeast, and even tobacco plants. Just more almost trial and error, I assume, and making sure that the compound is not toxic to the plant itself.
Yeah, the scope is almost pretty unlimited. I mean, the research project I did was about how they transfected about 10 genes that were not native to tobacco into tobacco. So that was 10 new genes into tobacco to make a product which tobacco had never made before, which happened to be a cancer drug. So you are really limited only by your imagination and the time you want to spend thinking into trying to get these genes to successfully work together. Yeah, if you can make milk, which is a mammalian product, in a bloody plant, I don't think you can really, feel like there's a limit on this. As long as you know the genes to make the thing you want to make, and there isn't some, if I don't really want to get too complex, but with tobacco, for example, when they were trying to make this pathway, sometimes tobacco's native enzymes made other little things that they weren't expecting, little byproducts. If soybean has particular enzymes like that and you're trying to make a thing, and soybean has a particularly strong pathway that then pulls all your product away, you might fail to make this thing, but then all you can really do is upregulate these genes until there's so much of this substrate that you're bound to get what you're looking for. So as I said, you're limited by your imagination.